Dear students, good morning. Today we are going to deal with the topic modified Mendelian ratio. In last 3 or 4 classes we go through Mendelian ratios. Different Mendelian ratios such as monohybrid cross and dihybrid cross. And in monohybrid cross you know that the ratio is 3:1 and in dihybrid ratio the ratio is 9:3:3:1. After that we go through what is back cross and also we go through test cross. We also go through the ratio of test cross and test cross of monohybrid and test cross of dihybrid. That all related with Mendel's work on pea plant, that is Pisum sativum. Now this another topic. Now modified Mendelian ratio. In Mendel's work what we see is that all the characters are controlled by a gene, and that gene has two alleles, a pair of alleles. One is dominant and other is recessive. The dominant one, when it is in the homozygous or in the heterozygous, presence of this dominant gene express the dominant character. But when the recessive allele is present, if it is in a homozygous case, that character will only express. If the homozygous allele is present along with the homozygous recessive, or no, the recessive allele is present along with the dominant allele, what is the morphology? You already know that in Mendel's work, the morphology or the phenotype is the dominant character. So, in Mendelian ratios, the dominant allele express the character and the recessive one will be suppressed by the dominant allele. Okay. Next, start here. Next, we are going to deal with the topic Mendelian ratio as allelic and non-allelic. What is the difference between these two? Allelic interaction means if the allele of the same gene interact. So, in non-Mendelian ratio or modified Mendelian ratio, there will be two types of interaction. One is allelic interaction and another is non-allelic interaction. In allelic interaction, the alleles of the same gene interact. But in non-allelic interaction, the alleles of two different genes interact. But when we deal with the two different allele interaction, we go through the dihybrid ratio. There will be two characters will be there. But what is the specialty of there? That two characters will not interact each other. But so here in non-Mendelian ratio, the two alleles interact each other. The two allele means the two alleles of two different genes interact each other. That is the main difference between allelic and non-allelic interaction. So students, once again, allelic interaction means alleles of the same gene interact. But in non-allelic means alleles of two different genes interact. We can cite it with the monohybrid ratio. Here in Mendelian ratio, if the alleles of the same gene is there, that alleles never interact. But in non-Mendelian ratio, if the alleles of same gene, it will interact. Here we can cite with the example of dihybrid as non-allelic interaction, that is interaction between two different genes. Okay. Let us go through some of the types of allelic and non-allelic interaction. The allelic interactions, 2 or 3 types we can go through. One is codominance. Then we can go through another one is multiple allelism. Then there will be codominance, multiple allelism. Then will be over dominance, etc. Then examples for non-allelic interaction as one is duplicate gene interaction, supplementary gene interaction, then inhibitory gene interaction, then epistasis. These all terms I am sure that these are not familiar with you. You are always familiar with the monohybrid and dihybrid ratios of Mendel. Once again go through the examples of allelic interactions. One is codominance, multiple allelism, overdominance and so on. And for non-allelic interaction, duplicate gene interaction, supplementary gene interactions, inhibitory gene interaction and epistasis and so on. These are the examples for allelic and non-allelic interaction. Among these, we have to go through each and every one as examples for studying modified Mendelian ratio. Okay students, today we can go through the allelic interaction. One example as incomplete dominance. You have to identify 1:2:1 as the ratio. This ratio which is similar to which ratio? Monohybrid genotypic ratio, isn't it? What is the phenotypic ratio? Monohybrid 3:1. This is the same as that of the genotypic ratio, but not we can go through incomplete dominance. Examples for studying this incomplete dominance are Antirrhinum majus and Mirabilis jalapa. This Mirabilis jalapa is the 4 o'clock plant and Antirrhinum majus is the snapdragon plant. We can go through examples of 4 o'clock plant. When we look into the 4 o'clock plant, there are two types of phenotypes are noted on flower color. One is red flower color and another is white flower color. We selected these two as parent. We can call it as the first or we can call it as P1 or the first parent. And when we cross these two plants, what is the progeny obtained is pink in color. One who studied the Mendelian ratio expects the phenotype as red. But when these two plants are crossed, they get pink colored flower. That there starts why these pink color express they are very surface and they go back to study detailed on these plants. It is found out that this is a heterozygous. Even though it is a heterozygous with dominant color, it cannot express the dominant character as complete. It cannot express the dominant character as complete. The dominant character has or dominant allele has some effect by the recessive allele. So here again we can find out that even though it is in the heterozygous condition, here the allele interact each other. What we noticed in the Mendelian ratio is that even though this F1 is, this is the F1. Even though this F1 is in heterozygous condition, what will be the expressed character? The expressed character will be that of the gene of that of the allele of the dominant one. Even though here it is in the heterozygous condition, the two alleles, the dominant and recessive allele interact each other and get a color which is a intermediate between this red and white as pink color. So they find out that the red flower color is with the capital R, that is homozygous, and white flower is with the recessive one as small r, and then the pink as capital R and small r. What they again do is that they raise F2 generation. How they raise F2 generation? They selected capital R, small r plant and another with capital R, small r, that is pink with the pink. Here what they get? Here capital R, small r. Here again capital R, small r. By checkerboard or by any other means we can write it as capital R, again capital R, small r, capital R, small r. We can find out as red, these two as pink and these as white. So the ratio we can write as 1:2:1. That is 1 red, 2 pink and 1 white. So here the ratio is 1:2:1. In this way the gene will be expressed. So here even though the F1 is heterozygous, the color is pink. When they crossed such two pink progeny as the second parent to raise the F2, they've identified that in the F2 generation they get the progeny in the ratio 1:2:1. Okay. Today I am winding up as here the pink color is not the complete expression by capital R. There is a interaction of small r gene product. What is the product? It's protein. So here in incomplete dominance there will be no complete dominance in F1. What will be there? There will be interaction between capital R and small r. So students, you go through the example. Once again, this is the allelic interaction. Here the alleles interact. What are the alleles? Capital R and small r interact. And its example is incomplete dominance. While studying this incomplete dominance, two plants are which will show this incomplete dominance. One is Antirrhinum majus, which is snapdragon, and another is Mirabilis jalapa, which is 4 o'clock plant. Okay, student, the ratio you also remember 1:2:1 as the incomplete dominance ratio. Thank you. Okay.